All right, I'm recording. Let's get rid of the science. All right, so let's go over vocabulary. Everybody's attention needs to be on the board. So, you won't understand meiosis and what's happening with meiosis if you don't understand the chromosome vocab. So let's look at this first one. Are you serious? I hate it when that's why I don't like to link up to it wirelessly, because it always drops. Oh. 1 second, y'all.
1.
That's all I need in my own way trying to be fun. Alright, let's get back to chromosome vocab. So, first slide. What type of chromosome is this? We talked about 2 types of chromosomes. Chromosomes can be duplicated and they can be unduplicated. So what kind of chromosome is illustrated on the board? Is this a duplicated chromosome or an unduplicated chromosome?
Unduplicated.
It's unduplicated. This is an unduplicated chromosome. This is what chromosomes look like before they enter S phase of a cell cycle. All right? Now, next slide. This is a chromosome immediately after the S phase. And what kind of chromosome is this?
Duplicated.
Duplicated chromosome. Notice the difference between an unduplicated chromosome and a duplicated chromosome. Are we paying attention to the board? Yes, ma'am. Right. This is before S phase. And what happens during S phase? DNA replicates so the chromosome can duplicate. Because what is a chromosome? A chromosome is a strand of DNA wrapped around protein. So if I need to make a copy of this chromosome, I have to replicate the DNA first. Then the chromosome can duplicate. Here, I have 1 chromosome and 1 chromatid. Here, I have 1 chromosome, but 2 chromatids, okay? Still 1 chromosome. It's called a duplicated chromosome. All right? What are these things right here called?
Chromatids.
What kind of chromatids? Are they sisters or are they non-sisters?
Sisters.
They're sisters. These are identical sister chromatids, okay? These are sister chromatids. Now, this is showing chromosomes that carry similar information but come from a different source. So one of them comes from mom, so it has a maternal source. The other one comes from dad. It's a paternal source. They carry the same type of information though. So what do we call these? What? Well, yes, they are non-sisters, but what are they called because they carry similar information?
They are called.
Genes are on the chromosome, but what are the chromosomes called? Because they carry similar information. What's that prefix I told y'all means same? So these are what? Homologs. Okay? They're homologs. These are homologous chromosomes because they carry similar information. Now this is a pair of unduplicated homologous chromosomes. These are duplicated homologous chromosomes. See the difference?
Go back.
Unduplicated homologs, duplicated homologs. See the difference? Now, what did I say this little thing right here is called? A chromatid. So I have a chromatid on this side in this homolog and a chromatid from this side in this homolog. So what kind of chromatids are they? Are they sisters or non-sisters?
Non-sisters.
Non-sisters. These 2 green ones are sisters to each other. But if I was to take a green one with a purple one, they are non-sisters. That's important for meiosis because during meiosis, to increase genetic diversity, the non-sister chromatids cross over and exchange information. So this purple one is going to pick up a little bit of green, and the green one is going to pick up a little bit of purple. Okay? Now this has to happen in the same area. Because remember, what's on these chromosomes?
Chromatids.
Genes. There's genes on the chromosome. So I can't take a piece from down here and put it up here, and a piece from up here and put it down here, because then I'm messing up the genes. Okay? The closer two genes are on a chromosome, the more likely they are to stay together. We call those linked genes. They tend to stay together during crossing over. That's why most people who have red hair also have freckles, because red hair and freckles are close together on a chromosome. The further two genes are from each other, the more likely they are to get separated. So if this was A, B, and C, during crossing over, A and B are probably going to stay together and go over here together, but the C is going to be left behind. Do you understand what I'm saying? The further apart they are, the more likely they are to get separated during crossing over. Now, when my two homologous chromosomes line up side by side, they form something. What do they form? What is it called? A tetrad. That is correct. They form a tetrad. Tetra means four. Tetra means four. Hey Parker, how many chromatids do I have up here? Four. 1, 2, 3, 4. 4 chromatids that carry similar information. So that's called a tetrad, okay? They have to line up side by side to cross over. Because if this was a chromosome 1 and this was a chromosome 2 and they crossed over, we have problems. Because that means you're putting chromosome 2 genes on a chromosome 1 and chromosome 1 genes on a chromosome 2.
That's called problem.
That's called a problem. That's called genetic disorder.
What's that actually make? It's like deformities?
Yes, that's a disorder that's called a translocation, because the genes change location with the chromosome, and there's several different disorders and cancers that can be caused by translocation.
Oh.
All right, now, when you got a question?
Oh, yeah, I was like, it's like cleft palate.
Don't know. I'm not sure about that one. I have to look. I don't know the exact cause of cleft palate. All right, when a tetrad crosses over, oh, I gave the answer. When a tetrad exchanges genetic information, this is called what? Crossing over. Now, the formation of the tetrad and crossing over happens during prophase 1. It happens during the first division of the nucleus. All right, we have two types of chromosomes, autosomes and sex chromosomes. Which ones are the non-gender determining chromosomes?
The first 22.
Autosomes. Which ones are the gender determining
The
chromosomes?
6 chromosomes.
What are the two sex chromosomes found in humans? X and Y. What's a genetic female?
XX.
What's a genetic male?
XY.
All right. Now let's talk about the stages of meiosis, okay? So the purpose of meiosis, we talked about last class, is for sexual reproduction, and it increases genetic variation. Hey, Muhammad, get up for a second.
Hey, everybody.
Pull the board up towards you and then unloosen. We're going to flip it over. We're going to flip it. Okay, lefty loosey, righty tighty. So turn it to the left.
Oh, yo.
There you go. Tighten it back up. Awesome sauce. I'm going to need you again one more time after this. Okay?
That's it.
Yep, for right now.
I thought that I was going to introduce myself.
Everybody know Muhammad. You're universal. All right, so meiosis, okay? Now first let me talk about mitosis. Mitosis has interphase, and then a division of the nucleus, and a division of the cytoplasm. So it's I, P, MAT, C. The division of the nucleus is the PMAC, prophase, metaphase, anaphase, telophase, okay? Meiosis divides the nucleus and the cytoplasm twice. So, interphase is interphase. It doesn't matter if it's mitosis or if it's meiosis. Interphase is interphase, and it only happens once per cell cycle. A cell will only replicate its DNA and duplicate its chromosomes once per cell cycle. Doesn't matter how many times it divides the nucleus, it only does interphase once. The three subphases of interphase are G1, S, and G2 in order. period of growth, where the DNA is replicated so the chromosomes can duplicate, and followed by a second period of growth. Interphase is interphase. Does not matter. Y'all with me on that?
Yes, ma'am.
How many times does a cell replicate its DNA in mitosis? What about meiosis? Once. It only replicates DNA one time because DNA is replicated in interphase. How many times does a cell do interphase in mitosis?
One.
What about meiosis?
One.
Once. Okay? Make sure you understand that. Meiosis 1 is the first division, first time the cell divides. Remember, a cell divides its nucleus first and its cytoplasm second. Meiosis 1 is the most important for genetic diversity. In meiosis 1, and you do have to put the 1s and 2s. So you have prophase 1, you got the PMAT again. Prophase 1, metaphase 1, anaphase 1, telophase 1. And then you have cytokinesis 1, which is sometimes called interkinesis because it's between the first and second divisions, okay? You may see it called interkinesis or you may see it called cytokinesis 1. They mean the same thing. Now, the stuff that happens in prophase of mitosis happens here as well, but then there's some extra stuff. So the nuclear membrane disappears. The centrioles migrate to opposite ends and form the spindle fibers. The spindle fibers connect to the chromosomes. All of that still happens. The difference is a tetrad is going to form instead of them getting in a straight line. Okay? So they're going to have a tetrad that forms, which means side by side, right? Those homologous chromosomes get side by side, and they do that so that the non-sisters can exchange genetic information. So the tetrad forms so that crossing over can occur. Crossing over is where you get your genetic diversity. Crossing over makes sure that no two chromatids are identical anymore. Remember those identical sisters we had when they replicated? They're not identical anymore. They're still sisters because they're still held together, but now instead of being identical twins, they like fraternal twins. They're not identical anymore, okay? Now, metaphase 1, the tetrads are going to line up, and I'm going to show you an illustration of this when I flip this board over, in the middle of the cell. Anaphase 1, your homologs separate. This is different from mitosis because in mitosis, wake up love, wake up love, wake up love. In mitosis and anaphase, the sisters separate. Let me see everybody's hands up. Do. One finger each. Everybody. If you in this room, I need to see your fingers move. All right. So, these are your chromosomes before S phase. During S phase, they replicate. So these are your duplicated chromosomes. In prophase 1, they form a tetrad so that they can cross over. Metaphase 1, they line up in their tetrad in the middle of the cell. Anaphase 1, the homologs separate. So now they're in two different cells, okay? They go through telophase, the cytoplasm divides. Each one of those different cells goes back through prophase again. Nucleus disappears. They go to metaphase, they line up. Here's a difference. In anaphase, this time, the sisters separate. Okay?
Huh?
Go back. Unduplicated chromosome. Make it sister. Show me prophase 1. What's this called when they get side by side? This is a tetrad. Alright, why do they form a tetrad? What's going to happen here?
Crossover.
For crossing over. Alright, now, show me, so metaphase 1, they line up like this, in the middle. They stay in the tetrad. Anaphase 1, say homologs separate.
Homologs separate.
Homologs separate. Now, these are going to go through the rest of the process, and these are now going to be in two different cells after cytokinesis 1. They go through prophase again, they go through metaphase again, and then they hit anaphase. Right now, they're still with the sister. It's not an identical sister, but they're still with their sisters, right? Anaphase means separate. So guess who's separating now? The sisters. All right? Show me metaphase 1. Metaphase 1. All right? Show me anaphase 1. Say homolog separate. Show me anaphase 2. Say sister separate. Sister separate.
Show me metaphase 2.
Well, it's hard to show metaphase 2 with just your fingers. Like, you can't, because, you know, you need to line up different stuff. So, yeah. All right. So, that's a big difference, because the purpose of meiosis 2 is to reduce the chromosome number. What's the purpose of meiosis 1? What's the purpose of meiosis 1? Genetic diversity. What's the purpose of meiosis 2?
Sister separate.
Reduce the chromosome number. Okay, let's get back. Let's try it again. What's the purpose of meiosis 1?
Genetic diversity.
What's the purpose of meiosis 2?
Reduce the chromosome.
Reduce the chromosome number. So in meiosis 2, so at the end of meiosis, you start off with one diploid cell. At the end of meiosis 1, you have 2 cells, but since no 2 sister chromatids are identical, they're considered haploid because you only have half of one of each of those chromatids. You don't have 2 of them because they're not identical anymore.
They crossed over.
And then each one, they're not identical because of crossing over, and then each one of these divides so that at the end of meiosis, you're going to have 4 haploid cells that are non-identical. Whereas mitosis, you start off with 1 diploid cell, you end with 2 diploid identical cells. See the difference?
Yeah.
How many cells do you end mitosis with? Are they diploid or haploid? Are they identical or non-identical? All right. How many cells do you end meiosis with?
4.
Are they diploid or haploid?
Haploid.
Are they identical or non-identical? The way I like to remember it is I look at the numbers. In mitosis, I'm going to have 2 that are 2N, and these 2 numbers are identical.
So it's diploid.
All right. In meiosis, I'm going to have 4 that are 1N, because remember the ones are understood, and those 2 numbers are non-identical.
Huh? Oh.
Okay? Y'all with me?
Yes.
If that'll help you remember it, might help somebody.
Well, look, this is how I remember that. Haploid.
Oh, recording. All right, so, Muhammad, come help me flip the board.
Haploid stands for half, so I'm saying half is haploid and it's for meiosis.
Right, but that's for the chromosome number, not the number of cells that you make, so I don't want you to get that confused, okay? Because you make twice as many cells, but you have half as many chromosomes. You ready?
Yeah.
Did you unscrew it?
Yeah.
Lefty loosey, righty tighty.
Oh,
Lefty loosey, righty tighty. All right. Y'all don't have to endure me trying to draw this like I did in 5th block.
Because we all have Muhammad.
No, because I already drew it. Help me push the board back.
You should have gone with it.
All right, so this is comparing mitosis to meiosis, okay? We're going to compare, and sometimes you need a visual to kind of help you see it. So, interphase is interphase, so interphase is not up here. We know what happens during interphase. Let's start at prophase. Prophase of mitosis. All right, now the nucleus disappears, so that's the dotted lines to show that the nucleus is disappearing. The spindle fibers form, and they attach to the chromatids. Notice that a spindle fiber is attached to each side of the chromatids, okay? During metaphase, they're going to pull those to the middle of the cell, and they're going to line up the chromosomes in the middle of the cell. During anaphase, who separated?
And the sister
The sister chromatids separated. So that in telophase, two new nuclei can form and you end up with two identical cells. Okay? Now, but let's look at the first division of meiosis. What's the first thing you notice that's different from the chromosomes in meiosis? What's the first thing you notice? They're crossing over, so they formed a tetrad, but also look at how the spindle's attached. Are they attached to both sides? Are they attached to every chromatid or just one chromatid in each chromosome? Right. They're only attached to one chromatid, one side in each chromosome. Look at the difference in the lineup. How many lines of chromosomes do we have in mitosis?
4.
There's one line.
Oh yeah, 2.
How many lines do we have in meiosis 1 though?
2.
There's 2 lines, okay? Now, let's look at anaphase 1 versus anaphase of mitosis. Who separated in anaphase of mitosis? The chromatids, the sisters did, but who separated in anaphase 1?
The chromosomes.
The homologous chromosomes did, okay? The homologs separated. So the chromosomes were not split, like the actual one chromosome wasn't split, it just separated from its homologue, okay? Now, telophase happens, cytokinesis happens. These two cells go directly into the next division. There is no interphase, okay? These two cells divide their nucleus again. Now, so back to prophase 2. Nuclear membrane disintegrates. Spindle fibers attach. Look at how the spindle fibers attach to chromosomes in prophase 2 and in prophase of mitosis. See how it's similar? Because the fibers are attached to both sides. When the fibers are attached to both sides of one chromosome, it's because the sisters are going to be separated. Okay? In metaphase 2, they line up in one line.
Just like the
Anaphase 2, the sisters are separated from each other. Meiosis 2 is most like, oh, about to kill myself.
Okay.
Some of y'all will be happy.
I wouldn't go that.
JC couldn't hold his, he was ready for it. You laughed or I had to point you out? Oh, she ready for, like, she probably calls it. She would come up here and trip me if I didn't fall. So we already know what Araya gonna do. All right, so meiosis 2 is most like mitosis, okay? When it comes to how they line up and how anaphase occurs. Are y'all with me?
Yeah.
Are you sure? Telophase, the nuclei form, cytokinesis, division happens. I end up with 4 cells because I have 2 cells that divide.
Okay.
All right?
Can I take a picture of this?
I'm gonna, the pictures will be, you remember how last time when I drew stuff, I took a picture and put it in Canvas?
You did?
Yeah, I did. So this picture will be in Canvas along with the PowerPoint. Okay? In Canvas, on the front page where it says slides and pictures. The pictures will be right there. All right.
A picture of me?
Now, let's go through the PowerPoint real quick. So, first of all, on your page 207, you're asked to draw a tetrad crossing over. You're going to draw homologous chromosomes lined up, and you're going to draw homologous chromosomes separating. So you have 3 boxes on that page. You may want to use a pen and a pencil, or 2 different colors. Yeah, I know I got color pencils over there in that white thing about the microwave if you want to use those. But go ahead and complete page 207 and page 208. Go ahead and do that.
Man, I'm getting old.
And because I know that, some of you guys don't understand my artistic disability, as it was called. So here is a slide showing a tetrad and crossing over right here, just in case you don't understand what I did. Hey Cam,
Yeah.
Cam, stop the camera.
Stop.
Yeah.